Hello and good day. On behalf of my co-authors, Mr. Ivan Culaba and Dr. Joel Maquiling, I am here to present our study on the fabrication and applications of a novel and multi-feature spectroscope. My outline of presentation will be as follows. First, I'll give a brief introduction to an optical device known as the spectroscope. Next, I will walk you through the major steps in fabricating the spectroscope and acquiring the emission, transmission, or absorption spectra of different samples. Then, I will highlight some notable results of our study, including the emission spectra of two light sources and the transmission spectra of thin green leaf and a dissolved chlorophyll sample. Finally, key takeaways will be articulated in the summary and conclusion. The main objective of the study is to develop a locally produced teaching instrument using indigenous materials as promulgated by the Philippines Enhanced Basic Education Act of 2013. In particular, this research aims to fabricate a multi-feature spectroscope that allows simultaneous observation of the emission spectrum of a light source and the transmission or absorption spectrum of transparent solid and liquid samples. In general, a spectroscope is an optical device that can stimulate students' interest in the visible light's constituent colors. This educational tool is conveniently used to qualitatively observe the emission spectra of various light sources. The basic design of a spectroscope, as shown in this figure, consists of a tube or box with fixed caps at both ends. At one end of the tube, the cap has a slit where the light enters. The cap on the other end of the tube has a grating over a small hole for observing the spectrum. Through the objectives of our study, we were able to form a research question. Particularly, we wish to investigate how can a simple spectroscope be modified to allow the simultaneous observation of the emission spectrum of a light source and the transmission or absorption spectrum of transparent, solid, and liquid samples. In this study, we used the commercially available cylindrical coin banks made of hard cardboard and with black plastic end caps. The leftmost figure shows two sample coin banks with their opposite end caps. The magnetic sheet with a slit hole is later on mounted on the topmost cup. Hence, the rightmost figure shows the topmost cup with the magnetic sheet, and the other cap with the grating mounted over the observation port in the form of a small hole. The magnetic sheet placed at the entrance slit provided useful features not found in a typical simple spectroscope. As shown in figure A in this slide, razor blades can be attached to the magnetic sheet to vary the slit width. Furthermore, we note that in figure B, a thin green leaf mounted with two steel strips can be conveniently used as a sample for observing transmission spectrum. And as shown in figure C, a cuvette with steel brackets can be easily attached for investigating transmission or absorption of colored liquid samples. In this study, we demonstrated the capability of the fabricated spectroscope by acquiring the emission spectra of various light sources. I will be presenting 2 out of the 4 light sources considered in this research. Shown in this slide is the emission spectrum of the compact fluorescent lamp, or CFL, as viewed from the spectroscope and recorded by a mobile phone camera. Note that the fluorescent light gives discrete Roy G. Biv lines as confirmed by the spectrometer data shown in the rightmost figure. Meanwhile, the emission spectrum of a helium discharge lamp in this slide shows its unique fingerprint lines of red, yellow, green, and blue as shown from right to left. The helium discharge lamp emits more prominent discrete lines as confirmed by the spectrometer data and also shown in the rightmost figure. Note that the shaded area in the spectrometer data are the only ones recorded by the mobile phone camera. In this research, the acquisition of the transmission or absorption spectra of a thin green leaf and chlorophyll extract highlights the integration of concepts and principles in physics, chemistry, and biology. It can be used later on as a springboard for multidisciplinary discussion on the subject of photosynthesis. In this figure, the upper half of the spectrum image shows the visible spectrum of sunlight, while the lower half is the light transmitted through the thin green leaf. Based on this figure, it is evident that the red and blue colors of the spectrum have been efficiently absorbed by the green leaf, consistent with previous studies involving photosynthesis and chlorophyll. In this slide, as shown in the leftmost figure, observing the transmission or absorption spectrum of the chlorophyll extract allows for a complementary discussion on which component of the green leaf absorb the said colors. Note that this spectrum is very similar to the one obtained from the experiment involving the thin green leaf. So the topmost graph on the right shows the absorbance spectrum of chlorophyll dissolved in 70% isopropyl alcohol, which is obviously dominated by the blue and red colors of the spectrum. Corresponding transmittance spectrum is also shown in the rightmost figure, consistent with the lower half of the spectrum as viewed from the spectroscope and recorded by a mobile phone camera. From this set of data, we can conclude that a multi-feature spectroscope was fabricated with a magnetic sheet, which allowed the slit width to be easily adjusted and conveniently hold transparent solid and liquid samples. The spectroscope was used to observe the emission spectra of different light sources. Furthermore, the device was utilized to simultaneously observe the emission spectrum of a light source and the transmission or absorption spectrum of solid and liquid samples. Finally, we note that this optical device is useful for both students and teachers in the STEM education subjects such as physics, chemistry, and biology. Finally, we would like to note that this work was supported by the Action Research to Innovate Science Teaching, or ARTIS project, which was co-funded by the European Union under the program Erasmus Plus, Capacity Building in Higher Education. Special thanks also to the Ateneo de Manila University for the conference subsidy. Here are the references highlighted in this presentation. Thank you for listening. We are now open to any questions and comments that you may have. Thank you very much.